Hello guys, welcome back to my channel Anytime Medicine, and today we are going to talk about is DNA. So first thing we are going to talk about is what is DNA and what function it has. It contains genetic code for developing, growth, reproduction and function of all non-living organisms and certain viruses that are existing in world. It is nucleus of eukaryotic cell and it is a cytoplasm of prokaryotic cell. Eukaryotic cell like our human cells and prokaryotic cell like bacteria. So now we are going to see the structure of DNA. It has two polynucleotide strands and they are composed of simple monomer structures, nucleotide. That nucleotides have sugar, nitrogen base and phosphate bonds. That sugar is ribose and that is backbone of DNA. Other structure is nucleoside and that only have base and sugar that doesn't have any phosphate group in it, like you can see in the picture. There are two types of nitrogen bases, purines and pyrimidines. You can see the structure of adenine and guanine. There are purines. And there are pyrimidines, cytosine, thymine and uracil. You can see the structure of cytosine, thymine and uracil. I have talked about purine and pyrimidine synthesis in my previous video and the drugs and disease related to it. You can check that out for better understanding of pyrimidines and purines. In order to synthesize DNA, nucleotides have to undergo phosphorylation, and that the process is phosphate group is being emerged in nucleotides and making the structure that can be used in DNA. You can see the pyrimidine, cytosine, thymine and uracil making cytidine, thymidine and uridine by undergoing phosphorylation, and the purines, adenine and guanine by undergoing phosphorylation, and they are making adenosine and guanosine, and those cytidine, thymidine, uridine, adenosine and guanosine contribute themselves in order to make the DNA. Now we are going to see nucleotide. It is synthesized as monophosphate and then converted into triphosphate form, and that form will added into DNA. If we take example as adenosine monophosphate, that will undergo phosphorylation and will create adenosine triphosphate, and that will go further process in making of DNA. In order to bind with another nucleotide in DNA, nucleotide have to detach two molecules of phosphate from the structure. You can see the structure of adenine monophosphate that has been already dephosphorylated. That means that it has already attached two molecules of phosphate. In this structure you can see the structure of hydroxyl, and that will attach to another nucleotide with the same strand which is down below it, and you can see the structure of oxygen group that will attach to another nucleotide in the same strand that is above it, and you can see the structure of nitrogen base, and that will attach to another nucleotide in the opposite strand with the another nitrogen base. So on your left picture you can see how they are binding with the another nucleotide in the same strand with the help of oxygen group and hydroxyl group. On your right picture you can see how they are binding with the another nucleotide on opposite strand with the help of nitrogenous base. Now we are going to talk about is DNA methylation is a biological epigenetic process in which methyl group added into DNA molecule, and that will modify the function and expression of gene. Adding methyl group in cytosine, we can see that only in human occurs in segment with CG patterns, where you can see lots of CG patterns, or you can see the CPG islands, that where the DNA methylation most probably to occur in both strands in DNA, and this will repress the DNA transcription. Approx 70% of all DNA are methylated in human body. If there is high amount of unmethylated DNA floating around in human body, that will trigger the immune response, and that immune response will destroy the unmethylated DNA. You can see the structure of cytosine that is being methylated into methylated cytosine. You can see the 1CH3 group in methylated DNA, that is what called methylation. So we have talked about DNA methylation in human. Now we are going to talk about is bacterial DNA methylation. Bacteria methylate both cytosine and adenine unlike human. In bacteria, methylation protects bacteria from virus which enter their unmethylated DNA. Unmethylated DNA destroyed by endonuclease, and this method is called restriction modification system. In human, they are destroyed by immune response, and in bacteria they are destroyed by restriction modification system, which is endonuclease. So further we are going to see is chromatin and nucleosome. The first chromatin is made up of DNA and proteins and is found in nucleus of eukaryotic cells. Chromatin get condensed into chromosomes, like you can see in a picture, chromatin getting condensed into chromosome. Inside chromatin there are units of proteins and DNA, like we have seen in the previous slide, which proteins are histones. DNA will wrap around the histones and will make nucleosome, like you can see in a picture that nucleosomes are being formed by DNA getting wrapping around the histones. If we talk about histones, histones are peptide-like structure. They have certain types like H1, H2A, H2B, H3 and H4. Except H1, all will wrap around to DNA. They contain high content of amino acid like lysine and arginine, and they are all positively charged and binds to negatively charged phosphate in DNA. Like we have seen in previous slide, there are certain types of histones. H1 histone is not found in nucleosome core because this is larger and more basic type, and that will be outside of the nucleosome core and bind with each other, and it will condense the DNA. Like you can see in a picture that histones are getting condensed into nucleosomes. So now let's see drugs induced lupus. In this patient have fever, joint pain and rash after starting certain drugs like hydralazine, procainamide and isoniazid. In this patient will have antibody against histones in most of the cases. If we have antibody against histone, we cannot condense the DNA, and therefore we can see the symptoms like joint pain, fever and rash. Note that classic lupus there will be antibody against double-stranded DNA, which is anti-double-stranded DNA antibody. If we see epigenetics in simple term, histone acetylation will increase the transcription because it will more open up the DNA, and DNA methylation will decrease the transcription, and because that will more tighten up the DNA. Now we are going to see histone acetylation. Like we have seen that histone has lysine and arginine, so there will be acetyl group added into lysine, and that will relaxes the chromatin for transcription. In deacetylation there will be reverse effect from the acetylation. So now we are going to see the histone deacetylase inhibitor, which has potential therapeutic effect and used as anti-cancer drug. Some cancer increases expression of these deacetylase enzymes in some tumors. If we give them this deacetylase inhibitor, that will have no effect in tumors. If we talk about Huntington disease, this is a disorder of movement that will have abnormal Huntington protein, and this protein will have functional mutation, and that will lead to histone deacetylation, and that will lead to gene silencing, which is a suppression of transcription, and which will lead to neuronal cell death in striatum. If we give them histone deacetylase inhibitor, that will inhibit the process of histone deacetylation in this gene and will suppress some symptoms of this Huntington disease. Thank you everyone for watching this video. Do subscribe my channel, share it to your friends, and see you until my next.